Good afternoon, everyone. This is Jay Scott here. Thank you all for being on this webinar today. I'm excited to present one of the Alex's Lemonade researchers here today. Who we have on the line is Will Parsons. And Will has been a long time grant recipient from Alex's Lemonade. He started off as a young investigator with us when he was at Johns Hopkins University. And then he went on to win one of our most prestigious awards, which is an A award, a 3-year, $125,000 a year award when he was at Texas Children's Hospital. Now Will is a member of our scientific advisory board. And just a little background on Will. He earned his PhD and his MD from Ohio State University. Then he went on to do his residency in pediatrics and his fellowship in pediatric hematology oncology at Johns Hopkins. His primary research interest is in the development of large-scale genomic methods to identify and evaluate cancer-causing mutations in human tumors. But we're real excited to have Will on this webinar today, and I'm going to switch things over to Will now. Will?
Yes.
Sorry, I had you muted, and you're unmuted now.
Great. Can you see my screen? I just want to make sure that everyone
I can.
On. You can?
Yep.
Great. So I'll just go ahead?
Yep.
Okay. So it's a pleasure to have the chance to talk to everybody for a couple minutes. As Jay said, I'm I have long time involvement with the foundation. I've been lucky enough to be funded by them through the course of my career. And what I'm going to talk about really briefly here today is some of the research, some of the research that I have been working on, which is to try and figure out how to use genetics and genomics to improve the care for kids with cancer, and how that has been funded by Alex's Lemonade Stand Foundation. Try and show kind of my research as an example of the sorts of projects that we can do, and very importantly, the ways in which the funding from an agency like the foundation can really make a big difference in the career and research plans of an investigator. So go to the next slide. For this audience, I'm not going to go into any great detail about the rationale for why we want to study pediatric cancers. It goes without saying for this group, but suffice it to say that, number one, there's still a number of cancers that we just don't do a good job of curing. For all the recent progress in terms of treating leukemias and some types of tumors, there's still some, and this is an example. This picture here is from a patient. It's called a pontine glioma or brainstem glioma, and this is the sort of tumor that I see this MRI scan in my clinic, and I know what's going to happen with this patient, and there's nothing that I'm going to be able to do about it. The second challenge is that some of these treatments that we have, even if we can cure patients, they have a lot of side effects and toxicities, and create a lot of challenges for our patients after they've been treated with our therapies. This slide, the second photo here, is an example of one of those. It's called an ependymoma. It's a more benign, so to speak, brain tumor, but it's one in which it can be cured with surgery, and potentially it's this white thing in the middle here, surgery or radiation therapy, but by doing that, we can cause neurocognitive problems for the kids. We can cause all sorts of deficits in terms of their ability to think and do and live as we'd like them to. So for even some of the ones that would on paper be cures, we have a big challenge. And the third is we're trying to figure out ways to detect tumors earlier. So knowing, for example, kids that are at risk of developing tumors, and also being able to early on detect them at the time when they're still treatable. And so that's what we're working on. And the way that I've chosen to focus on this is by learning about the genetics of tumors. Basically what this slide shows, it shows the progression from a normal cell, a cell that's going to develop into a tumor, going through all the various stages that it goes through until it gets to a metastatic cancer. So one that can spread throughout the body, and one is as bad as it gets. And there's been a lot of research done showing that mutations and alterations and changes in various genes in the tumor, so the DNA of the tumor is affected, can lead to progressing from a normal cell to a cancer cell. And so there's been a lot of research over the last 10, 20, 30 years trying to look at some of these individual genes. KRAS is one of those examples, and try to figure out how we can use this to improve our ability to diagnose patients, and how we can design drugs to actually specifically fight against or target each of these mutations in the cell. So if what was once a normal cell now has a mutation in this gene called KRAS, which causes it to grow and spread and do all the bad things that cancer does, to come up with ways to actually find a drug that treats specifically that mutation or that gene and not one that's as toxic on the children. And if we're talking about single genes, then we're talking about genetics. Genomics really just is a matter of scale. So it's looking at the genes and the genetic causes of tumors, but it's looking at all of them and not just a single one. So one thing that's also very important in pediatric oncology is we know many of these mutations that occur in tumors and cause them to grow are not in the normal cells of the child, that there's something that's happened in that particular cell and then it's caused that cell to grow into a tumor and be out of control. But it's also true that there can be mutations that are inherited or mutations that are new as the child is being being born, that can put them at risk of developing other cancers. And so for adults, we know some of these are mutations in BRCA1 and BRCA2 predispose adults to getting various types of cancers. And that's even more important in kids. And this is a fancy family tree or pedigree essentially showing one of our new patients. It was a 2-year-old child who had a neuroblastoma. And at the same time she was being treated, her mother was also being treated for a cancer, in this case a breast cancer. And each of these other circles or squares that has either a green or a yellow chunk in it are all patients in that family with tumors. And so we know there's some cases where there's obviously some familial component to this. And so the focus of my research is try to learn about what are the inherited risks and things that cause children to get tumors, and then why do they develop from there. And fortunately, over the time when I've been focusing on this topic in the lab and being funded by the foundation, we've gone where the technologies are completely different than they used to be, where someone like me used to spend a career studying one particular gene and trying to figure out what it's doing in cells and how it might be related to cancer. Now we can be using these so-called whole genome or exome or genomic technologies to look at all of the different genes in the cell. So the technologies have allowed us to do experiments that we never could have done previously. And the goal of my research is to take this sort of knowledge, so learning about the genetics and genomics of these cancers, and then actually translating into improved patient care. Obviously, we're very interested in learning about the biology of the tumors and how they're forming and why, the basic science. But as an oncologist, and specifically a neuro-oncologist, a brain tumor doctor, I'm much more interested in the clinical application and trying to figure out how to use this information to diagnose our patients, give them better prognostic information, and then treat them and do that in a better way. So that's the goal of our research. The one project that I'm working on at the moment, which has kind of evolved out of the ALSF Lemonade Stand Foundation projects, is a project of this type, and that's to take these sort of methods, so these genomic sequencing methods, from the laboratory to the clinic, and then use them actually within the clinical care of our patients to provide information that's reliable, that can be obtained quickly enough that we can help figure out better ways to take care of them. And not just the patient, but also their family in cases where there's inherited risks of cancers and other children and adults might be affected. So really what we want to do is we want to have a patient have surgery. The pathologist will look at that this is a medulloblastoma, a certain type of brain tumor under the microscope. But we don't want to treat those all in one way. We want to use a molecular classification system such that we know for each tumor what's the specific biology driving that tumor, what are the specific mutations driving that tumor, and then give different treatments. So the first step in that, and applying these clinical genomics, is cancer gene discovery. And these were the sorts of projects that I was first funded by the foundation. So really trying to look and say, okay, in pediatric brain tumors, for example, what are the important genes? What is the important biology? So many steps away from using it, but just trying to learn. And this is a slide showing just an example of one of those types of mutations we found. Gliomas are a common type of brain tumor in adults, the most common malignant brain tumor. And through a project partially funded by the ALSF Lemonade Stand Foundation, we found mutations that in this gene called IDH1 and IDH2. And there are mutations that affect kind of the action or the money part of the protein, so to speak. This is a slide showing a picture of the protein. And what they do is they affect how this protein binds to the substrate, the thing that it acts upon. And the mutation makes it such that there's a totally irregular function here and it causes tumors. And the interesting part is from the time when we first reported these findings, and these are just photos of the first page of some of those papers back from a handful of years ago, so September 2008, February 2009, with research that was partially funded by the foundation. These things can then be translated into huge amounts of progress and huge amounts of change. So one project identified one set of interesting genes, these IDH1 and IDH2, and suddenly it's a whole new area of cancer research. So when I just did a search yesterday when I was preparing the talk, in our PubMed, which is where we look up all our scientific literature, and I just searched IDH1 and cancer, those two words, and 563 research papers and projects came up that had followed up on these sort of findings. So this is a way in which funding a project with some interesting findings can then be amplified and translated. And there are people doing all sorts of things, from very basic science to figuring out how to use this for imaging and taking better pictures and MRIs of patients, to designing inhibitors of this actual compound or of this actual molecule so that we can treat our patients better. And so it's really amazing the progress actually that can be made with these sorts of things. So that was earlier in my career. Later in my career, so to speak, work that's also partially been funded by the foundation, we are trying to take these methods and not just use them to identify genes that are important for in the laboratory, that we can study them there, but actually come back to the clinic. And so this is a project that I'm going to talk about. It's called the Basic 3 Project, and it stands for Baylor Advancing Sequencing into Childhood Cancer Care. And it's funded by the NHGRI, or the National Institutes of Health, specifically the National Human Genome Research Institute. And it's a project that I'm partnered with Dr. Sharon Plan, who's one of the cancer geneticists here at Texas Children's and also Baylor College of Medicine. And she also happens to have had research in this area funded by Alex's Lemonade Stand Foundation. So it's really a family affair, so to speak. And the point of this project is to do this. We are taking all of our patients who are being diagnosed with tumors here at the hospital, at Texas Children's Hospital. So we anticipate not leukemia patients, but all the solid tumor patients, so the brain tumor patients and neuroblastomas and Wilms tumors and those sort of things. We're offering enrollment to all these patients on our study. We think it'll be about 200 children over 2 years. We get blood and tumor samples from them if they've consented to the study. We also ask for blood samples from the parents so we can start to learn about what the genetics are of the inheritance of some of these tumors. Those samples go to the laboratory, so not a research lab, a clinical lab, just like a laboratory for the hospital that does blood counts or a potassium level or those sorts of things. They give results back, so they actually generate clinical reports to say the germline is basically from the blood. These are the mutations that we see in this patient's blood that might explain why they have cancer. These are mutations we see in the patient's tumor, might explain why the tumor is developing and give clues about how to treat it. And then these results go back into the medical record. The EMR is the electronic medical record. They're shared with the patient's primary doctor, and the doctor then shares them with the family. And the point of this study is following these patients, taking care of them, following what treatments they give, and how the doctors use this sort of sequencing information is the point of the study. So this is how we think things are going to be, that we're going to learn about patients' tumors, for example, at the time of diagnosis, learn about their blood and see why they've developed some of these cancers if we can, and then treat them in different ways and use different medicines depending on what specific characteristic their tumor has. And so this is kind of the clinical games of the project. First of all, we really need to learn how to talk about these sorts of studies with our patients, because it's not as simple as just determining one result of one test. This is sequencing their genomes. This is trying to see what are all the risk factors for cancer, what are other things that they might have inherited that put them at risk for other diseases that we need to take care of. And we really need to know how to talk to families and parents and patients about that. And then the second is we're trying to see what is the actual utility of these results. So set aside kind of the basic science and how we can learn about each patient's tumors. We're trying to figure out how we can take these results and figure out what it means for taking care of that family. So for example, if we identify a mutation that's been inherited in a child that causes them to be at risk of developing other cancers, how do we take care of that child? How do we test for other children who might also be affected in that family? How do we look at other family members? And other questions such as, when we're looking to see cancer risk, we're also going to find other medically actionable things. So for example, if someone has inherited a mutation, there's rare diseases that cause heart conduction problems or that cause seizures or these sorts of things. If we find those, how do we use them to take care of our patients? And so those are some of the practical things. And then on the tumor side, we're trying to figure out, will the doctors actually take this information that says these are the particular mutations and genes that are important in your patient's cancer, and will that affect how they take care of them, what drugs and chemotherapy agents they give? And this is kind of the pipeline and how we think this is how it's going to be done in the future, this personalized or precision cancer care, so to speak. But we decided that just presuming that to be the case is not good enough, and we wanted to do a project where we actually do all the work necessary to do this kind of testing, to run clinical trials based upon this sort of information, and then study it for our patients. The parents of the patients are actually also subjects in our study, both because they give blood samples and also because one of the questions we have is, what kind of information are they interested in learning about when it comes to genetic tests, and how can we use this to make their care better? And so that's the project. In addition, obviously, we can use these same samples to do all sorts of more basic research. So we asked for additional patient blood samples, parent blood samples, tumor samples from if these children go back to the operating room at a later time, because we want to know all these other biological questions. For specific tumor types, what are the important mutations? What do we see in terms of changes before treatment or after treatment? Can we see signs that might explain why some people respond and when some people don't? These are some of the very basic questions we're trying to ask. So far, we've been enrolling patients since August of 2012. So far, when this slide was made, there are about 72 patients and families already enrolled on our study. And this is just a pie chart showing some of the common types of pediatric tumors. So CNS, central nervous system or brain tumors, neuroblastoma, Wilms tumor, et cetera. And the point of this slide is just to say that our families and the parents and our patients are very excited about doing this sort of work. They want to know more about their tumors. They want to learn more that helps other children. They want to get some clues about why the tumors might have developed in ways that we can better take care of them. And so the families of patients have been very enthusiastic about the study, even though it's genetic information. It's information that could potentially make some people anxious. We also ask if both for the blood samples from the various family members, as well as we kind of survey and interview folks at various time points. So it's something that they're really putting an effort to and contributing. And we're finding that the patients and families are very enthusiastic. And so, for example, these numbers on the outside are how many patients with each of these tumor types have already been enrolled on the study. As I said, we're about a quarter of the way there. We're shooting for about 280 patients over 2 years, which is roughly how many of these patients we expect to have. So here's an example of one of these cases. So it's the first patient. This is an example of how the tumor results can sometimes be helpful. So it's a 7-year-old boy. He came to the hospital with a belly mass. He'd been not feeling quite well for a month or two, had occasional vomiting and not feeling well, but it was difficult to diagnose what the problem was. Finally, he was noticed to have a big liver and actually a tumor that you could feel if you were palpating his belly. And so we had these imaging, and these are basic scans. The one's an MRI scan and one's a CAT scan. On the left is an MRI scan. And look at this. You can picture the patient lying on a table with their feet towards you. So the front side of the patient is at the top, the back side is at the back, and the right and the left, so to speak. And so this right here, for example, in white is the spinal cord, and this is the vertebrae. So this is supposed to be liver here, and this whole tumor, this whole whitish gray chunky thing is a huge tumor taking over a good part of his liver, which is bad. The other part was bad is that this, the dot is not where I want it to be, the red dot, this white part here, this is showing a metastasis or a bit of this tumor that has spread to his lungs already. So this is a patient that standard treatments are not going to be very good for. We already know it's a very high-risk patient. When the pathologist looked at the tumor under the microscope, and these are just pictures of the tumor from various magnifications, they couldn't tell us exactly what kind of tumor this was. So there's some cases where they could say it was a liver tumor, and they could say it was a cancer, but they couldn't tell us what type such that we could have a better idea of what specific treatments to give. And so in that case, what we do is the oncologist go ahead and treat with one type of treatment for a specific type of tumor called hepatoblastoma. But what happened was the patient progressed right through this tumor. And so this is the sort of situation where having some additional information about the tumor, about the genetics, about what kind of molecular pathways might be activated can give us a hint about what drugs to use. Because when we did, for example, the sequencing of this patient's tumor, we found there's a mutation in a gene called NRAS, and if you can see in the blue circle or blue rectangle here, that's a gene called RAS. This shows just a very key, very important pathway in cancer biology. So it's a pathway where it's supposed to work by various growth factors, activate the cell, they bind to some receptor on the surface of the cell, and then the signal goes into the cell to divide and grow and do all sorts of things. What happens in a lot of cancers is this circuit can be dysregulated. So in some way it's turned on such that it's never off, and it's just growing and growing and growing. And so this is an example of this particular tumor was mutated, had a mutation, had an error in a specific gene called NRAS, a gene called RAS. And the question is then how can you use that information to try and take better care of patients? And the example is there are now inhibitors or drugs being developed to each of these in these blue circles and yellow circles. These are other genes in the pathway. To each and other members of the pathway, so PI3K, AKT, mTOR, MEK, ERK, these are all different genes and the specifics are unimportant. But the fact of the matter is there are now drugs being developed not just to take care of all cancers, but to specifically target molecular pathways in the cells that are turning an individual patient's cancer on. And so in this case, for example, there are drugs targeting RAF, for example, one called sorafenib, and it's an oral agent that can be given and is FDA approved and available. There's also another drug called rapamycin, which targets this gene called mTOR. And so this is a case where there's no good inhibitor of this gene called RAS, but what we can do is try to use drugs to some of these downstream components of the pathway to try and keep this signal that's coming in, this mutation, from actually having an impact on the cancer cell. And so in this case, the patient was actually treated by his oncologist with a combination of several drugs, sorafenib and rapamycin. And it turns out that mutations in this gene called RAS are characteristic more of the adult type of liver cancer, which is hepatocellular carcinoma. And so he's treating the patient more in that fashion. So it's just an example in theory of the ways that we can learn something about a patient's tumor, especially in cases where we don't quite know what to do, and then try to use some of the new medicines that are available to learn from that information. The other example of how we can use this is in terms of inherited risk of cancer. So this is another patient of ours. She's a 14-year-old girl who had a glioblastoma. It's the most malignant, the worst type of brain tumor, essentially, in kids and adults. And when we talked to this family, they didn't really mention a family history of other family members having cancer. And so this child was being treated. She had a surgery and radiation therapy, and then was actually on a clinical trial. But then when we went to talk to the family about this genetic study, we actually found a little more information. So this shows the pedigree or their family tree. And so this is the child, the 14-year-old with the black arrow here at the bottom. And patients marked in blue are patients who've had cancer. And so her father and mother, both 43 years old, neither one had cancer. And so that, I think, was what our oncologist had asked the family at first, because none of the siblings, neither the two brothers had cancer, neither the parents had cancer. But if you went back far enough, so to grandmother who had a colon cancer, an aunt, great aunt who had a breast cancer, ovarian cancer, lung cancer, et cetera, there is a family history of the cancer there. And these are the sort of cases where it really would be good to know about this in advance, because we can do various genetic testing for other family members. We can screen to try and prevent or diagnose tumors early. And so what we found in this child, we found a mutation in a gene called MSH2, which is what's called a mismatch repair gene. It's involved in repairing errors in DNA. And so if it gets mutated or there's an error in it, it causes a risk of other cancers. And so it's actually associated with risk of colon cancer, gliomas, which is GBM, the type of cancer this child had, et cetera. So this is a case where we'll be able to take that, in addition to taking care of the child and trying to treat their tumor as best we can, we could also learn more about what the risks are for the family and other family members, and hopefully prevent some other children from being affected with these, or some of the adults with other tumors. So just like we can learn about the tumor genetics, we can also learn about the inherited genetics. And these are the ways that we're trying to figure out how to make use of this information. So just to give a little bit of background, as Jay mentioned, I was first funded as a young investigator back in 2007, I believe. It seems a long time ago now. And then I was lucky enough to also get an A award in 2010, and that sponsored some of the early work looking for some of these genes causing cancer, and then transitioning to some of these more clinical studies. For someone like me who's doing this sort of projects, you need funding from a variety of sources, and one great thing about getting this sort of funding from ALSF Lemonade Foundation is then it helps me get the credibility and the ability to gain funding for other projects from other sources. So not just from other private foundations, such as the ABTA, et cetera, but then actually moving down to what is sort of the gold standard for us, which is also obtaining funding from the NIH and from the government, which is incredibly hard to do at the moment because it's so competitive and for the financial reasons that all of you are well aware. But that's one of the goals, and so getting this sort of research support from the foundation, being able to start these projects, then allowed me to do work that I can continue on to NIH projects. Most recently, we actually got funded by the Stand Up to Cancer and the St. Baldrick's Foundation to do a project for the first pediatric cancer dream team project, so to speak, in collaboration with John Maris and the group at Children's Hospital in Philadelphia, as well as folks at the NCI. And again, Jay and Scott, Jay and the rest had some influence on this as advocates for the project, as trying to make the case that we need pediatric cancer funding for some of these groups. So in general, it's the situation that success breeds success, so to speak, or some of the funding that leads to gene discoveries and leads to early discoveries can then be translated all the way through. So this project is really especially important for researchers at critical career transitions, so to speak, and these are the places where people tend to go off from the career research track, where they get discouraged, they don't have funding, they can't do the projects, they can't hire the people they need to do them. And so, for example, going from when we're doing our clinical training as an oncology fellow to become a faculty member and getting your first job, having the example of an ALSF Young Investigator Award is critical, both in terms of allowing you to do the research, but then also giving you the credibility to show that you can get this sort of funding and that you'll get more of it. And then going from a junior faculty to an established faculty member it's also crucial. And that's the case where, for example, the A award can help, because it's a larger amount of money, it's more input, it allows you to hire some additional people, it really allows you to ask bigger questions. And these are the points where if someone who's trying to be a physician scientist can't get research funding, then they'll go back to the clinic and they'll do very good work there, but they won't be able to work in the lab to make some of this progress. And so that's the case where this really helps. It specifically provides a lot of things to research. One is power and credibility. So giving us the opportunity to ask the sorts of questions that we're interested in asking, to do the sort of work we want to do, and then have other people say, yeah, that's someone that, Alex's Lemonade Stand believes in them, and so therefore they must be good. And that really, it's impossible to overstate how important that is for us early in our careers. The next is it obviously encourages us and encourages research to do additional work. It also applies pressure, because in addition to each of us answering to our own conscience, so to speak, of really being driven to find results and to make progress, it's helpful to have foundations, to have other advocates, to have other people who are keeping our noses to the grindstone, so to speak, to make sure that they can encourage and help in any way they can. And finally, it provides hope, because these are the sorts of projects that we can do, and this is the sort of, even over just a handful of years, we can see both developments in learning about the biology of tumors as well as some of the clinical applications for them. And so with that, I'll go ahead and stop. So these genetics and genomics approaches are really helping us learn more about pediatric cancers. It's just one example of the type of research that the foundation funds. The results of these studies not only are important for the biology, but actually have real clinical applications and are helping us to try and study how to improve patient care. And Alex's Lemonade Stand Foundation, as I hope I've shown, really provides critical support for these efforts, which translates into exponential amplification of donations that are made and initial work that's done. And so I'll leave it there. This is actually a papier-mâché heart that my first patient ever made for me that I keep in my office and like to show in all my talks, because we don't want to forget who's really making this research possible and why we're doing it. So I'll leave it there, and I'm happy to take questions or go ahead with letting you all go about your day.
Thank you, Will. I really appreciate it, and I hope everyone found it useful. If you have any questions, you can type them into the question section on the right hand of your screen. I do have a couple here, Will, but I just would like to, again, thank you for coming on. And really, Will is a poster child for what we hope for without Alex's Lemonade Stand funding, where we were able to see Will grow from being a young investigator to the A award recipient to going and getting really big dollars from the government. And now I believe you are the co-director of the brain tumor or neuro-oncology program at Texas Children's, right?
Yes, yes. So it's fun to have that these are the sorts of lead things, these projects and this support that leads to opportunities to really make a difference both in the lab and the clinic. It's fantastic.
And also now Will and his colleagues at Texas Children's have taken on some Alex's Lemonade Stand summer fellowships for medical students and graduate students. And so they're trying to help us train the next generation of researchers. So the first question I have here is, in your opinion, do you think we will ever find new cures?
We'll absolutely find new cures. There are new cures being found all the time. Our dream that we might find one specific or like one overarching cure or one way to treat all cancers is clearly just a dream. But what we're learning as we learn more about the biology and of different tumors and how they're all not the same, even if they look the same under the microscope. We're learning about how different subgroups of the patients need different kinds of therapy. So just like each child is different, each patient or group of patients can be different. So I'm absolutely confident that we're going to make additional progress. We've really had an opportunity the last handful of years with some of these new technologies to learn incredibly more about some of these pediatric tumors in particular in the lab, that now over the next handful of years the pressure is on us to translate those to the clinic, and so that's what I'm going to focus on at this point.
So the next question is kind of an interesting one, Will, and it's, do you think enough young researchers are attracted to the field of pediatric oncology? And if not, what do you think are some things we can do about this?
My bias would be, it's a great question, my bias would be that we can never have enough young people interested in pediatric oncology, and that's why I actually think programs such as some of the early Alex's Lemonade Stand Foundation programs, such as the summer student internships and those sorts of things, I think can make an incredible difference. There's a lot of young people interested in the sciences, interested in biology, interested in the lab, going to med school, that some of them have an idea from early on exactly what they're interested in, what they want to study, but I think there are many more that are really just looking for an opportunity to do good, useful, meaningful research. And so hooking them early to get them interested in our patients and in our issues and problems, I think, is key. The summer student I had last summer, funded by the foundation, actually is now, he had not known that he was going to go to med school. He was a senior actually this year, and he's going to be spending a year in the lab, and then he's applying to MD PhD training programs because he's interested in learning more about, interested in becoming an orthopedic surgeon who helps take care of kids with bone tumors and learn more about those. So it can really make an incredible difference, like some early investment in those sort of people, I think.
Oh, that's great. So I got another question or another comment here that the person said I didn't need to read aloud, but I'm going to. I'm not going to tell you who it was so I don't embarrass her. But
Okay.
It says, thank you so much for a very clear presentation that will enable us to explain to others again why the support is so crucial for the incubator projects as well as the projects, wait a second, as well as the projects that are about to have a breakthrough. So I
think that's a great point. And I'm glad that I managed to at least convey that, because I really think, just like investment in the summer students, investment in the early projects, and the fact of the matter may be that some of those early projects don't lead anywhere. In fact, a decent number of them may not lead, when I say not anywhere, I mean not to leading to a cure, but they may lead to learn more about the biology of the tumor, and they may, in some way we don't understand, lead to further progress. But those are both in terms of investing in the people, getting them hooked in this area and getting them doing this sort of research, and in the ways that if we actually do find discoveries from those early projects, they can be incredibly, a dollar spent funding one of those projects can then, when you think about, if we added up, for example, from that IDH1 and IDH2, those gene mutations we found, if we added up the research funding that people had gotten to study cancers based upon learning about those genes, in both adults and kids, it would be many, many, many millions of dollars. And so it's a way that we can really, really take what the foundation is doing and make it even better.
So I have one last question here, and it is, how did you first hear about Alex's Lemonade Stand?
That's interesting. My memory is getting bad enough these days that I have a challenge remembering. I'm pretty sure that I first heard about it in relation to not the grant funding, but in relation to the the actual lemonade stands and the fundraising efforts and that whole cool part of it. And then later when I was starting to try and figure out, how I was going to support my research in pediatric cancer, and then I was lucky enough to come across or have someone recommend to me that the foundation would be a good place to start. So.
Isn't it a weird thing to tell people that your research is funded by a lemonade stand?
Yeah, it's great. It's actually, I think, my kid, I have 3 daughters who are typically not very impressed with anything that Dad does most of the time, but the fact that I'm funded by Alex's Lemonade Stand Foundation and that I have a little lemon on my, thing on my white coat, and at one point had some wristbands and such, they get a big kick out of that, and they think it's great.
So that makes you a little bit
really
cool?
Appreciative. It makes me just slightly cool, but not very.
Well, thank you so much, Will. I really appreciate it, and appreciate everyone getting on the call, and keep up the great work, and we're proud to be associated with you.
No problem. I'm happy to answer any questions if people have something they want to follow up with me later or something that I can help with. Please just let me know.
Okay. Thanks, Will.
All right. Bye.
Bye.